If you are just joining us or coming from one of the other breakout rooms, you're with Farming Today. Coming into the noon hour here, we have our final two presentations of the conference, which we hope you've enjoyed and had a great time or had as much fun as we have. Thank you to all of our speakers, all of our sponsors. So before we break out, because it is 11:59, but I wanted to say that thank you to our sponsors, Compeer Financial, Minnesota Milk, Central Minnesota Credit Union, and Centricare. They were our commodity sponsors for this event. So if you are...
Good morning. I think we're ready to start this session. We're asking people to stay muted unless they have questions, and Anna, our speaker, can tell us if she'd like to take questions along the way or wait till the end and how to deal with those questions as we go along. But we generally would like to stay muted unless we're asking a question. You can also put questions in the chat box as we go along here too, that we can watch and take care of along the way. So please welcome Dr. Anna Cates, the State Soil Health Specialist at the University of Minnesota, who is here to tell us about how soil health practices can build carbon and other benefits. One of the questions I asked about Anna in this carbon sequestering kind of a question is, how do farmers see a benefit this? Maybe the soil's happy, but how does it benefit the farmer and the farm family in their efforts to make a living and as well as to take care of their farms? So Anna, I'll turn it over to you, and we look forward to what you have to share today.
Okay, thanks, Dan. Right, so I'm going to I know there was just a presentation from the Nature Conservancy on carbon markets and kind of this big picture interest in carbon. I'm going to back up a little bit and talk about the science of how carbon is stored in the soil, because I think understanding that from the mechanistic level can help us understand why certain practices are promoted at the state level. So I'll talk about those things, talk a little bit about function, and and you can certainly write questions in the chat. I'll do my best to monitor it, or do the equivalent of jumping up and down, waving your hand by unmuting yourself and saying hello. I don't have my contact information on this slide, but I'm pretty findable at the university. That's catesa@umn.edu. Feel free to write me if anything comes up. So I start with this definition of soil health, the continued capacity of the soil to function as a vital living ecosystem, because I like that it focuses on functions, and that leaves the definition flexible to the functions we care about most. The functions that we care about are going to vary depending on the plot of land we're talking about. You know, a stream bank, we might want different functions than we want in our yard or than we want in the middle of a crop field. A pasture versus a annually cropped area. So I think it's really relevant to think of soil health as something pretty flexible that can mean different things to different farmers, different things to different stakeholders, etc. It does make it a little bit complicated and a little bit wishy-washy. It's not a spring nitrate level. It's something a little bit more complex and subjective than that. I think a flow chart like this, though, can help us conceptualize what we mean by soil health, though. In this case, it's just a series of questions about functions that the soil might have. And if you answer on the positive side, then you can say your soil is probably healthy. And if you answer on the negative side, you can say your soil is probably not healthy. So if your soil is blowing or flowing away, it's probably not healthy. The baseline to having healthy soil is keeping it in place. The others have to do with water there in the middle, and I'm going to talk a little bit more specifically about that. And then the last two have to do with annual crop plants. Does your crop recover most of the nutrients that you apply? Or are there areas where plants die or grow poorly? And I like to include those in this flow chart, which I took from an extension agent out in Washington, but I think it's relevant here too. I think that it's important to include plants as an indicator of healthy soil, because a lot of times plant function is really one of those primary functions we care about. And looking at where you have problem areas in terms of your crop health, crop yield, crop emergence, things like that can also be indicators that there are soil problems. So I'm going to get in a little bit more to water here. There were a couple of functions listed around water there, and these pictures kind of illustrate essentially situations where you're ripe to impede water infiltration, because on the right, you can see you've got a wheel track where water is just going to flow right down, and it'll probably leave something like the picture on the left where you've got a crusty surface. And in both cases, water has a hard time infiltrating in that situation. We actually need a pretty diverse set of functions from the soil regarding water. This picture shows two different soil structures interacting with water. And I think that water is important to focus on because we've had some very wet years, and so water management has been very much on people's minds. And also because of this scenario where we actually need so much from our soil in terms of how it behaves in relation to water. So here you can see on the left, a less aggregated soil with smaller particles. And then, so these top views are looking down at the surface of the soil. On the right, you have a well-aggregated soil with larger particles. And so you can see there's different sizes of particles. There's a little residue there. The bottom half of the figure shows a cutaway of the soil. So if you've dug a hole and you're looking right at the soil surface, and the blue lines represent water trying to vertically enter from rain. Maybe some of you are looking at rain outside your window like I am. This is happening as we speak. On the left, you can see that the rain is attempting to enter between small soil particles, but there just aren't very many pathways for the water to get in. And that's going to obviously slow your infiltration. On the right, you have more effective infiltration and drainage when you get a lot of water at a time, when you have large macropores between aggregates. So it's just like you can imagine a room full of beach balls has more space between the particles than a room full of marbles. That's the situation you have in the soil with either less aggregated soil or more aggregated soil. The other thing you get from soil aggregates, if you zoom in, is inside the aggregates, you get some pore spaces. And so that means that you get the second function that we really care about with water and soil, which is water sticking around for a long time. So not only do we need water to enter the soil, drain through the soil as quickly as possible when we get a lot of rain, but we also need water to stick around for when it's dry. This might be one of those years where the water storage is more important than the water movement through the soil. And really, it's only with soil aggregates that we're able to get both of those functions from our soil. So the next thing I'm going to talk about is how soil aggregates form and the impact of different practices on those aggregates. So soil aggregates form from soil organic matter. This is a visual of the soil food web, vastly simplified. I'm not going to go over all of these particular creatures in the soil, but the important thing is that there's a transformation of plant carbon into the bodies of microbes, the bodies of larger organisms like mites and nematodes and millipedes. And that transformation of carbon creates the soil organic matter that sticks around in the soil for a long time. Some of the carbon is transformed into carbon dioxide and released. And in the carbon sequestration conversation, we talk a lot about minimizing that release of soil carbon. But I think it's important to acknowledge that we're never going to get down to zero in terms of carbon release from the soil, because in order to build carbon, you have to support these organisms that eat and breathe carbon just like us. You know, we're all going to go eat lunch here pretty soon, and afterwards, you know, we're going to use carbon energy and breathe out carbon dioxide. And that's how microbes work too. So when you look at your percent organic matter in the soil, it's not just a flat quantity, it also represents a lot of different processes and a lot of different organisms kind of carrying out their life cycles. The next part of keeping soil organic matter around in those aggregates has to do with the surface area of the soil particles. So in here, this picture, I've zoomed in on the surface of a clay particle, and you can see this is just a tiny, tiny scale, 5 micrometers. A micrometer is 1,000th of a millimeter. And in those little layers, that's where organic matter is able to stick around. That's where water is able to be held. And essentially you get clay covered with a sticky surface of organic matter, of residues from the microbial bodies, byproducts when they die, their excrement, all that kind of thing just sticks around on the soil surface. That snowballs into this larger level of organization, which is aggregates. So if you can imagine those sticky clay particles building like a snowball, with this heterogeneous mix of organic matter of different sizes, living things, and also some space for water and air inside those aggregates. This cartoon shows you how a largish aggregate, and keep in mind this is still less than 1 millimeter wide, but what we call a large aggregate in the soil science world has within it smaller aggregates, which have within them those clay and silt particles covered with organic matter films. They're held together with these stringy things like fungal hyphae, the red and yellow lines in this picture, and also the green plant roots. So you need the organic matter to build the aggregates. And then once you have aggregates, that protects the organic matter for a longer period of time, because you might have organic matter kind of stuck in here that's not accessible to a microbe that's over here. Microbes are aquatic organisms that can move. Some of them can move, some of them can't within a water film. But if they're physically isolated from organic matter, they're not likely to be able to break it down and release that carbon dioxide. There's a positive feedback here. You need the organic matter to build the aggregates. And then once you have the aggregates, you're able to protect the organic matter or the carbon for a longer period of time. I do use organic matter and carbon somewhat interchangeably. Organic matter is generally about 50% carbon, and they're just two different methods of measuring something similar in the lab. Carbon tends to be a little more precise, so scientists tend to use carbon. Organic matter is cheaper to measure, so farmers tend to use organic matter. This is just an overall sketch of carbon that I'm not going to talk about today due to time. My computer's frozen for a minute, so hopefully we'll catch up here. But I wanted to get now into what this means on the landscape. So we want to build carbon on the landscape. What are some ways in which we could do that? Or I'm also going to talk about what are the ways in which our practices affect our ability to build carbon on the landscape, and in particular with soil structure and aggregation. So these are the soil health principles that are often used. Each one is essentially working together to build carbon in the soil by protecting soil structure. Starting off with keeping the ground covered, just on that premise, I already stated that if you're losing soil because it's blowing away, it's not protected, you aren't going to build soil or organic matter or carbon. Minimize disturbance. Those stringy fungal hyphae and roots are really vulnerable to tillage disturbance, as I'll show a little bit more data on. And so if you have a lot of disturbance events, you're going to build less carbon or organic matter in your soil. Keep living roots in the ground. That's because it's not only dead plant material that feed that food web, but living roots slough off carbon that's very easy for microbes to digest. And so microbes live right on the roots of plants, eat carbon there, and have essentially a symbiotic relationship where they are getting some energy from the plant carbon, and they're also breaking down nutrients, organic forms of nitrogen and phosphorus for plants to take up. So having those living roots there really supports that food web, probably more than anything you leave on the surface. Diversification above ground leads to that food web having a more diverse menu, essentially, of carbon options. And again, if we're thinking about keeping them robustly supported, that can be helpful. Integrating livestock, the last principle, I'm not going to talk about a lot here, but I will say that it comes in two ways. One is the manure. Manure is a great carbon source for that food web. It's really effective at building structure, and it's a special kind of quality of carbon, if you will. The second thing about integrating livestock is that it really makes the first 4 pieces more easy to integrate on a farm system. So if you're thinking about trying to keep living roots in the ground year round, you're thinking, well, What am I going to do with something with a perennial in the rotation? You know, am I looking at alfalfa? Am I looking at a small grain? And those crops are more profitable when you can feed them to livestock, obviously. So integrating livestock should be thought of in both like the biophysical way, and that it does contribute to the soil food web, and it really a lot in the farm systems way, because it really changes sort of the economics and the logistics of incorporating those other principles, and in particular, a diversified rotation that includes perennials, includes winter cover crops, etc. Okay, so now I'm going to talk about a couple specific practices that fit within those principles. I'm going to talk about tillage, I'm going to talk about cover crops, and talk about what happens to soil biology, soil structure with both of those. So with tillage, as I said, this really is the worst for the large aggregates. The small aggregates that I've described as a snowball of sticky clay and organic matter, they're more robust to tillage. They're not likely to be broken down. But here what they show is the percent of water stable aggregates at different size classes. So the largest aggregates here, the ones greater than 2 millimeter, and for a visual, your pencil lead is like 7 millimeters. So a 2 millimeter aggregate is certainly something you can see, but still small. And you can see that in the first 2 bars, those are the no-till system, they have more of these big aggregates than in the second 2 bars. That's the tilled system. The other treatment in here was stubble or no stubble, and that seems to have an effect too. So if you have both no-till and you leave your stubble on the surface, which is probably feeding the microbes that build that carbon and build aggregates, that's going to be more effective than if you are all the way down here at tilled without stubble. And you can see, because it's not that the soil is necessarily blowing away, but it's just essentially in a smaller size class. So if you can go back to that first visual I showed you of the water trying to enter between tiny particles, in these systems where you don't have the stubble, and you have, especially with that stubble, then you just have more particles that are smaller. See, because you have less food for the microbes, less organic matter building up and making those sticky aggregates. So instead, you're left with these tiny particles that are probably going to reduce your infiltration overall. The other piece here is showing how fungi in particular are affected in tillage and fungicide situations. And so here they compared both no fungicide to fungicide in a tilled scenario, the pinkish bars, and fungicide to no fungicide in a no-till situation. And here the metric is grams carbon in large aggregates per kilogram of soil. But the take home is just the title here, that fungi build large aggregates. So if you have a no-till system where fungi are allowed to thrive and you're not applying fungicide, you're going to have they're going to have more capacity to build those large aggregates than in the other scenarios described here. And then this is a really cool situation where they tilled a native prairie system for just one time. And this is at, I think, 0 to 7 centimeters is what that's about. So they tilled a system just once, and then they compared in dark the never tilled system to the tilled once system. And over time, you can see this was after the first tillage event in the summer of 2002, the mean weight diameter of aggregates just drops instantly in that system that experiences tillage for the first time. Then it builds up a little bit over the next 2 years, but it's not really getting back up to this initial level. So this mean weight diameter indicator essentially means that there's fewer large aggregates after a tillage event, and it stays that way for a long time. So while I agree with my colleague Jody DeYoung-Hughes that reducing tillage at all is always a positive sign for your soil structure and building organic matter, it's true that even a little bit of tillage is going to destroy the aggregates, and it takes time to recover. Unfortunately, with soil, and this is true with organic matter in general, it's easy to destroy the structure and lose organic matter, lose stability, and it's hard to get it back. Okay, so next I'm going to talk about some biology and structure pieces with cover crops, really more biology with cover crops and more structure with tillage, but they intersect. So this is some data I took looking at different cover crop systems in both a grain and a silage corn system, where we had either a bluegrass or a rye cover crop. And the metric we were looking at here was potentially mineralizable carbon, which essentially measures the amount of carbon dioxide produced by that food web. And in a grain system, the food web was not more active with the cover crop. But in a silage system, it really was. And keep in mind that with that silage system, you're removing so much residue that the cover crop represents a really important input to the microbes at a time when they're not getting any other inputs. And so I think that that gets back to this, concept of feeding the food web with your farming practices. And if you're feeding that food web, then they're able to build the structure and also break down nutrients and make those available for crops in the coming year. This is from a meta-analysis, which means a study of many studies looking at different microbial responses to cover crops. The top half looks at the total microbial biomass, which was increased with cover crops, meaning that all of the dots are to the right of this dotted line. On the bottom part, they looked at the microbial respiration, which is the same thing I looked at here, the carbon dioxide respired by microbes. And they've measured different kinds of cover crop, a legume species, a grass species, mixed, or some others. And in all cases, they found that the cover crops just increased the overall microbial population and also increased their respiration, which can be seen as a measure of activity. This last study I'm going to talk about gets at, it's brand new. I just saw it last week, and it gets at this question of whether having cover crops in the mix can kind of change your long-term farm forecast. And so this figure, they looked at just a cover crop versus no cover crop system at 3 different landscape positions. So summit, back slope, and toe slope. And then the 2 colors represent the cover crops. And there's 2 pieces of information on this square graph. One is annual yield. So if a cover crop had increased the yield at the back slope position, for example, we'd see a movement from left to right in the position of this dot. And there is a little bit of movement. So maybe here on the slope, cover crops increased yield a little bit. Whereas here at the summit, that's pretty marginal if there is an increase at all. But what's really striking is that at every landscape position, cover crops reduce the yield variation. So in all cases, the green dot is substantially below the brown dot. And this gets at sort of the larger question of cover crops as part of a system which is going to increase your resiliency to different kinds of weather years. It's going to maybe increase your yields where they are more marginal rather than in the areas where they're already, where your yields are already really good. So the idea is that it can essentially make up for poor conditions in other ways, perhaps. And then I'm going to skip this study, but if you're interested, the takeaway is that there is an increase in infiltration with cover crop. Because I want to talk a little bit about the larger question about cover crop benefits. This is a figure from the Ag Evidence Dashboard, which I really recommend to anybody who wants to see what is the science, what data do we have in the Midwest around the effect of cover crops or other practices on different metrics of interest. I pulled out a few here that they have some of the most data on. Each one of those lines represents a study done in the Midwest. And if the dark bar is in the green area, that means that the average change when adding cover crops was positive. So in some cases, like nutrient runoff, you want that change to be in the negative direction, but it's a good thing, right? We want less nutrient runoff. Same thing with nematode or pathogen pressure. That would be you want to be lower in those categories. Whereas in something like carbon storage, you would like to see it going up. So what I want you to look at is essentially that in some cases, like soil structure and carbon storage, we're seeing kind of, gosh, that is above the line, but there's a lot of studies where we found that carbon storage went down with cover crops. And we have a lot that are pretty close to zero where carbon storage didn't change very much with cover crops. Same thing with soil structure. In cases like nutrient runoff, maybe, or nematode pressure, we're actually seeing like a more consistent benefit overall. And then in this case, we're seeing a more consistent issue with pathogens. And pathogens is a wide category. I think you can dig into that and see some more specific things if you're interested. But I want to put cover crops in context as something that might provide any number of benefits besides just carbon in there if you implement them on your farm. However, what does it look like when you implement cover crops on your farm? I'm not going to talk a lot about the technical details right now, but just to say that the amount of biomass you get from a cover crop might vary substantially. This is just some research data showing 2 different years, 3 different locations. I'm not highlighting any crop or system in particular, but just to show you that, you know, sometimes we're way down here and sometimes we're at like 4 times the quantity of biomass produced by cover crops, even at one location, and at same seeding types and other systems, seeding timings and other systems. So, and these pictures are representative of what that looks like on the ground. So these numbers are all in kilograms per hectare, but it's pretty similar. This is actually 500 pounds per acre and 1,000 pounds per acre and 2,000 pounds per acre. But I put the units on top that are relevant to these ones. So you can see that our research trial cover crops, you know, at the best, they were hitting this 1,000 pounds per acre type metric, but they were a little bit off of that. More often they were below that. So you're going to get a different result in terms of how your cover crop affects your field, depending on how much biomass you're producing. This figure just shows that. And I guess I want to emphasize that I think there are some pieces that we can more reliably count on with cover crops. For example, infiltration and soil biology, soil nitrate scavenging, then perhaps soil carbon. We know that this works over time, but it can kind of go very, very slowly, very, very incremental, and maybe hard to measure. And it'll be very much related to how much biomass you get. So a few take-home messages: you need your biology to build soil structure and carbon. Tillage is going to disrupt structure, disrupting that cycle. Cover crops feed biology, which can build structure to some extent, but it's going to be slow on the carbon front. If you implement both together, I think it's a case of 1 + 1 being greater than 2. For one thing, your cover crops are a cost to the farm, but reducing tillage can be a cost saving. So together, this can be an overall economic win. And together, they can also speed soil development faster than either one by themselves. I'll just end with my contact information here. I encourage everybody to just do visual assessments of their soil out in the field, especially getting out after a rain, maybe a day like today where there's still a little frost in the ground. It's interesting to see how fast you have infiltration in some areas of the field versus others. You can monitor your organic matter levels, but keep in mind those are likely to change slowly. And really, when you engage in soil stewardship with an eye on soil health, you're really engaging with this long process of soil formation. The soil we're working on is formed after glaciations that we really can hardly imagine. They were tens of thousands of years ago. This soil was used by Native Americans in different ways. It was burned, it was grazed. White people have been farming it for a few hundred years. We've changed it dramatically, and we kind of don't know what's happening next. So when you think about soil health, I encourage you to think about it as an adventure, a long-term adventure, which you should figure out how it best fits into the logistics and goals of your operation. I will stop there. If we have time for any questions, I'll take them.
I'll ask one, Anna, to start off with. When I was young, back in my high school days, boy classes with my dad, I learned that plowing down corn stalks and some of those kind of things was really good for the soil, maintaining soil humus and all those good things. And then Jody, who you mentioned earlier, told me 10, 15 years ago that crop residue left in the soil surface does a lot more toward building soil and contributing to the things you've talked about than crop residue that's worked into the soil. And so, so can you comment on that? What that's true? What makes that true?
Sure. Yeah, there's pluses and minuses. When you incorporate corn stalks or any residue into the surface, you're making it get you're putting it deeper in the profile faster than it would by just percolating down from the top. You know, worms work pretty regularly, but they're not as fast as a chisel plow probably in terms of burying your corn stalks. Right. And so often in a tilled profile, we see a deeper level, high levels of soil carbon a little bit deeper than we do in a no-till soil profile. But when you're doing that, you're disrupting all that structure and kind of the natural process of carbon formation. And so you're going to plateau at a lower level of carbon, essentially, because you're constantly disrupting the potential of the soil to store it.
All right. I think she said something, if I remember right, I maybe don't remember right, that a lot more of the carbon when it's worked into the soil or buried in the soil, it gets given off as carbon dioxide because it decomposes faster than it would if it's left on top. Was that part of it?
Yeah, sure. And it also decomposes carbon that was otherwise going to stay protected because you've broken up aggregates that you weren't going to break up otherwise. So carbon that was going to be protected might be lost. Yeah, residue management is a real issue. And a lot of I didn't, like I say, get into the technical details of how to implement this stuff. But when you're going to a no-till system, thinking about it the fall before you start is critical so that you don't have a wet mat of residue that you're trying to no-till through. Keeping more vertical residue in the form of raising your chopping head is really important for a successful no-till system, especially in Minnesota.
All right. If there are any other questions, you're welcome to turn your mic on and ask them or to put them in the chat line. And we can take other questions that you might have. I think about the residue thing partly in terms of, then I think about, okay, so what do we do with manure? So we want to incorporate manure for capturing the nutrients and making them available to the crop. But if that's the same time we have that dynamic of some of that material would be from carbon building, maybe you don't get as much out of burying manure material that way. Especially, that probably matters too more if there's a lot of bedding in the manure compared to something that's mostly in a liquid stage.
Sure. Yeah, my colleague Melissa Wilson is doing research on that in a couple areas. First of all, really trying to understand the best ways to use low disturbance injection systems. And so I can't comment on the technical aspects of that, except to say that from a theoretical standpoint of protecting your soil structure, low disturbance is a good thing. And the other thing she's looking at is applying manure, sometimes with these low disturbance injection systems, on cover crops so that the nutrients that are released are ready, have a living plant there to take them up. That can reduce your risk of runoff and kind of change the calculus in terms of your nutrient management plan. I will say that maybe the gold standard for getting nutrients out on a crop field, or if you have a cover crop that then you can fence in and graze so that the animals are applying the manure themselves at a time of year when it's kind of, it's not as much of a hazard for water quality.
Right. All right. Any other questions from anyone? All right, another question I'd have, I'll just ask this and then we'll kind of give you a chance to get on with the rest of your day or see whether somebody else has one they want to ask. Related to the cover crops, you mentioned their value as being kind of neutral in terms of building carbon or not building carbon. And could you tell in those studies where you're looking at that, how that was factored by whether the cover crop was being grazed off or harvested off compared to whether it was all that material was left in the field? I know there's a lot of that plant material that's underground plant material compared to above ground that you would be harvesting.
Yeah, that's an interesting point. Certainly pulling off the above ground material would probably reduce your carbon sequestration. I think in most of those studies, the cover crops were not grazed or harvested just because that's not as frequently done. I think that the most important factor is how much biomass is produced at all, because of the below ground biomass is going to be there regardless of what you do with above ground management. And I think in the systems where you saw increases in carbon, my gut, they don't have this data all connected on that site, but my gut would be that those are areas where they had high biomass systems. That's been the case in research that I've done. If you have a good year for cover crop growth in the fall, then you are more likely to see a carbon boost that year. And all systems work that way. Some years are more favorable for building soil carbon than others. If you have a warm year with a more active microbial community in the spring, like maybe this year, you know, we might see less potential for carbon sequestration in a warm year like that.
All right. All right. All right. I don't see any other questions in the chat box. Anybody else want to speak up with a question? Give you a chance. All right. Anna, I really want to thank you for sharing the information that you shared today. And if people are ready to leave this room, you can go back to the, click on the leave room button if you're moving on with the rest of your day here. I think Laura plans to have some wrap-up comments as we wrap up our conference here for these last 2 days. And I think it's really interesting times with looking at what we're learning about soils and soil health and managing for the health of those microbes in the soil, as well as the health of the crop and what we have to gain from that. It's really intriguing times to be watching that. And it's really good to be observant out in fields and thinking about how you balance that, creating a seed bed and getting the soil warmed up for growing the crop, as well as taking care of some of these other things. And being good students of our land and what we observe in the field is really a key kind of a deal. So with that, we want to wish everyone also that they will have a safe planting season. And as you plan for that, we wish you the best in your discussion among your family members and workers on your farm to have a safe planting season and even an enjoyable spring planting season this year as well. So we wish everyone well through the growing season. Thank you for joining us for this conference. If you'd like to contact any of us with comments about it or feedback or suggestions for future learning, we're always interested in getting your feedback about these things. So thanks again, Anna. Appreciate your contribution today.
Thanks, everybody.
Okay, bye now.